are these different types of tissue and try to identify these type of tissues as we're going through this. I can't find the link to yesterday's lecture. Oh yes, Queen. The reason why it's not up yet, I'm having some issues with the recording. I actually forgot to record it using my own file, and so I'm dependent on the file that is recorded through the Zoom, and it hasn't been released yet. I've been asking the support to try to find an answer as to what's going on with that. Okay, so I'll try to get it up there as soon as possible. But yeah, it's not up yet. I'll try to post it up on YouTube if possible. Okay, so here, if we take a look at this here, let me go ahead and use it darker. Okay, so if I were to point to this right here, I say identify this tissue here. Once again, you want to be thinking about, okay, is there a lumen or a space here? If there is, then, okay, we know it's an epithelium. If it's not, then it's one of the other type of tissues. And so by using this, you should be able to see that there is a space here. There is a lumen over here. So knowing that, we know that this is some sort of epithelial tissue. And since we have this as an epithelial tissue, what you could see here is now you're going to be asking how many layers are there on this epithelial tissue here. Just by you typing in chat, how many layers do you think this is at the part I'm pointing to right now? How many layers? Yeah, a lot of you are saying there's one layer. You're absolutely right. There's only one layer here. Okay, so there's only this layer here. Now the next question is the shape. What is the shape of these cells here? We know it's one layer, but what's the shape here? Yeah, it's flat. Very good. So your complete answer for this will be that it's simple squamous epithelium. And you'll be right. Okay, so that's kind of why simple? Very good question. Simple because simple is only one layer. It's only one layer. So if it's only one layer, we call that simple. Okay. All right. So my next question will be identify an organ that has this tissue. Okay. So ID an organ that has this tissue. Let's see if you could put some of the answers in chat here. Kidney's a good one. Good. Any other ones? The lungs. Good. Good. Liver, not so much for this one. Liver, not so much. The alveolus, great. Okay, that's in the lungs. Good. Lining of the heart. If you could be a little bit... lining of the heart. The reason why I don't like that answer is because I got a feeling, I'm not sure if you actually know or not about that, you know, kind of thing. So I wouldn't put the lining. When you say the lining of the heart, can you be a little bit more specific? What lining of the heart are you talking about? The serous membranes. If you say serous membrane, awesome. Okay, I would totally accept that. If you say visceral pericardium, awesome. Parietal pericardium, awesome. You know, kind of thing. So I want you to actually show me that you actually know what you're talking about instead of just randomly guessing an organ, basically. On future exam quizzes, will you have arrows for the parts of the tissues you want us to identify? Yeah, it will be kind of like what I'm doing right here. I'm going to be pointing to a part and asking you identify this tissue or identify this organ. Okay. All right, Nicole, I will get to there. I will slowly get to there. So in a couple slides, I will get to there. Unless you're in a rush to leave, I will get to there in just a few slides. Yes, I am recording this right now. Okay, so if I'm pointing to, let me point to another one that's a little bit more obvious for now. If I point to here, and I say identify this tissue here, okay, what would be your answer kind of thing? Identify this tissue. remember, you'll be thinking about, okay, is there a space slash lumen, right, kind of thing. You'll also be thinking, if there is a space slash lumen, how many layers is this? And after figuring out, okay, if you know how many layers is this, what is the shape of the top layers of these cells here? So on and so forth. Well, let's take a look. Okay. There is a space here. Here's the lumen. It's not that clear, but there is a space there. And how many layers is this? If you look at this, I'm going to make it pretty obvious for you. How many layers is this here? Yeah, it's just one. Okay. Anybody have issues with that so far? Yeah, they look more round than cube. I totally agree with you. Okay. But remember, when I said that with cuboidal, is it going to be perfectly the same tallness as its width? No, not really, kind of thing. So it's going to have this cubish kind of shape. Okay. So yes, this is only one, so that's why we call it simple. And like the shape, like we were just talking about right now, is kind of cubish, right? So cuboidal. And since there's a lumen, we know this is an epithelium here. Any questions regarding two? So far, any questions? Simple squamous epithelium, simple cuboidal epithelium. That's okay if you have questions, I don't mind. But I'm just kind of wondering, getting a feel, or whether or not you can start recognizing this. So only epithelial tissue will have a lumen or space. Yes, for the generally speaking. The reason why I say that is because every so often there are going to be tears in the tissue, because these tissue, we're slicing them, right? When we slice them, they're not going to be perfectly sliced every single time. There will be some tearing. If there's some tearing, there's going to be a space and stuff like that. So you want to look at it generally speaking, overall. So you want to be recognizing this. Don't only rely on this picture of it. Go online and Google kidney slide or something like that, and look at the different kidney slide, or simple squamous epithelium. Look at the different types of simple squamous epithelium or simple cuboidal epithelium, and so on and so forth. That way, the more that you expose yourself to all these different tissues, the better you become at recognizing them. Okay. Are you going to show? It really depends. Like even here, right? If you look at it, even here, there is some tearing over here. Okay, so even here there's some tearing over here. So this is not epithelium. This is part of the areolar connective tissue and stuff. So it happens, it happens. So the thing is, like, I want you to get used to seeing all these different type of tissues and not have the perfect tissue every single time. Okay. All right, how would you know if it's a tear or space? The more you get exposed to looking at these things, the easier it becomes to recognize these things. That's what I'm trying to get at. Okay. All right, so far we have these two here. Okay, let me go ahead and go to the next slide here. All right, so if I was to put an arrow over here, and oh, by the way, let's go back actually. We didn't ask the follow-up question. Identify an organ that has this simple cuboidal epithelium. Thyroid, awesome. Kidney, awesome. Kidney tubules, I'll accept that. Yeah, good. Okay, good. All right. Renal tubules is the more correct term for that, but I'll accept that for now. Okay. So if I were to point to here, no, that's okay. Don't worry. I want you to keep on guessing. We're just learning through this process. Yeah, I know what you meant exactly. So here, if we take a look at this here, what type of tissue is this here? Once again, you're thinking about, okay, is there a space / lumen? Okay, if there is, we know what type of tissue there is. from this, how many layers is it? And then after looking at, how many layers there are, what is the shape of the top layers, and so on and so forth. Okay. So if you look at this, notice how all the nuclei here, they're all along the same plane, right? They're all kind of along the same plane. Yes, you notice that they're kind of moving up and down, but what I mean by that they're on the same plane, they are all kind of in a row. Yes, you can move it up and down, going like this, right? But still, they're kind of all in one row kind of thing. They're not stacked upon each other like this, right? Notice that? Do you see that they're not actually stacked? If they were stacked upon each other, there will be another nuclei up there, another nuclei down here, but they're kind of just horizontally with each other. Okay? Since they're horizontally with each other like this, then it is only one layer then. Okay? It is only one layer. So this is going to be simple. And then we look at the shape. The shape on this is going to be like this. Okay. So actually it's even taller than that. It's like this more so. I made it too short, sorry. Let me correct that. It's more like this shape. So on and so forth. Okay. Are the white spots goblet cells? Very good. Okay, so these cells here are the goblet cells here. Okay, very good. Are the very ends, the white part underneath the tissue, Meissner corpuscles? No, they're not. But I see where you're getting at. Meissner corpuscles are going to be found in the skin, though. But you're absolutely right. It would, if this was skin, it would be found in this area. Okay, good. But this isn't skin, though. That's the thing. So it's not in this case scenario. You have the right idea, though. So that's good. You have the right idea. All right. So this is simple. And because of its shape, right, you notice how it's very elongated. You had a question? Sorry? Or someone had a question?
Oh, sorry, I just logged on.
Oh, no worries. Okay. So this is simple. And because of its elongation, this is going to be columnar. And we know this is a lumen here. So this is an epithelium. Questions regarding to this? Here's another structures that you could see, okay? So if you were to zoom in, you could kind of see it here, okay? You see this little dark outline right at the edge over here? Okay, I'm going to zoom in to kind of help you see that, but you should be able to see even without it being zoomed. Do you see this little purple dark edge on the end over here? That's this one structure that increases the surface area for these cells to absorb things or secrete things. Anybody remember what those structures are? Yeah, these are the microvilli. Very good. Those are the microvilli. Okay, good. Okay. All right. So looking at this here, we know what tissue it is. What is the name and organ? It's hard to see the brushes. Exactly. That's what I want you to take out of that. It's going to be really hard to see the brush border because of how small they are. No, we're just going over different types of tissue, how to identify them, and identifying an organ that contains that tissue. Yes, so small intestine, great, will be a great example. This is actually from the small intestine here. Stomach will be great. Awesome. Any other ones that you could potentially list? Large intestine will be a good one too. Okay. You could say bronchioles or uterine tubes. That would be fine too. Would we have to specify if they're talking about ciliated or non? No, no, you don't have to. However, if you could recognize those things, they will help you to identify the tissue first of all. But second of all, depending on how I feel, there might be an extra credit question or two. So if you're able to identify these structures, you might be able to get extra credit for it too. Okay. Potentially, I'm not saying definitely I'm going to put extra credit on there. I haven't really determined about that, but at the very least, it's going to help you identify the organs. Okay. GI tract would be fine. Ideally, I'd rather have you actually say an organ kind of thing. Especially, I don't want you to say GI. I want you to actually spell things out instead of having abbreviations too. Okay. All right, so that was simple columnar epithelium. Okay, let's take a look at this one here. I know it's kind of sideways, and so you kind of have to tilt your head. Okay, so if I say identify this tissue over here, okay, I'll make it a little bit clearer here by just labeling it like this with a bracket. But what is this tissue over here called? I'll use a different pen because it's so dark. So if you take a look at this, okay, first of all, what general type of tissue? Well, there is a lumen here, right? And since there is a lumen here, we could figure out, okay, what type of tissue is this, right? Okay, I have an idea about that. Right? So yeah, we know that it's some sort of epithelial tissue. Very good. And now knowing that this is an epithelial tissue, now we gotta figure out how many layers is this? What is the type of shape of these cells? And so on and so forth. Okay. This is where you gotta start recognizing some of the other types of structures to help you identify what this is. Okay. You'll notice that the nuclei here, here's a nucleus here, here's another nucleus, here's a nucleus, here's a nucleus. You'll notice that it looks like it's stratified, right? Doesn't it? It kind of looks like it's stratified. The reason why it's not stratified columnar epithelium is I'm not going to test you on stratified columnar epithelium. It's one of our crossed out things. That's one thing, okay? And then there are other reasons behind it too, okay? So it's not the stratified columnar, okay? So these cells, I'm going to draw it out for you, okay? So this cell looks like this. This cell looks like this. Okay, this cell is like this. This is more or less like this. Notice how all of them are attached to the basement membrane here. So this, in actuality, is a simple epithelial tissue. Okay? It's the pseudostratified columnar epithelium. So you might be wondering, well, how would I know that? Well, first of all, like we said earlier, I won't test you on the stratified columnar epithelium. Okay, so that's one thing. Okay, here are some other things to identify this. Okay, this is the only organ pretty much that I'm going to test you on the pseudostratified columnar epithelium. And this one happens to have these structures over here. What are these structures here that you see on the very outer edges here? Okay, notice how they're pretty long compared to the previous slide that we had, right? The previous slide, if we look at it, look how small that is, right? It's so thin. You could barely see it, right? Kind of thing. But this one, you can actually see it. Granted, this is zoomed in, okay? Granted, I'll agree this is zoomed in. But even if we were to zoom in on here, right? Even if we zoomed in, notice how you could barely see that thin line of microvilli there, right? So cilia is a lot longer. It's a lot longer compared to the microvilli. And since we know that there are cilia here, and also there are these cells here, right? These, they don't stain that well, these cells here. These are the goblet cells. Okay. So now we have these other structures, other cells to help you identify this is pseudostratified columnar epithelium. Okay. Yes, there are other cells that are ciliated. If it was a ciliated simple columnar epithelium, you will not have this stacking of the nuclei. The nuclei will be all along the same row. Ali, you'll get more familiar. Just keep on looking at these things over and over again. I gave an example on my Wednesday class that if you ever have identical twins as friends and stuff like that, when you first met them, right, you couldn't tell them apart from each other. But as you continue to get more interactions with them over the years or time, you are able to recognize them and their subtle differences here and there. Same thing with this. At first, it looks all the same to you, but the more that you get exposure to this, the more you kind of quiz yourself, quiz each other, your study groups or your lab partners, send out one of these pictures out to them. Say, identify this tissue, identify an organ with that tissue, and quiz each other. The more that you do this, the better you become. Okay. What's after the basement membrane? Is that areolar connective tissue? That's correct. This is areolar connective tissue right here. Very good. That's areolar connective tissue. Okay, so going back here, pseudostratified columnar epithelium, the one I'm going to test you on will have cilia with it. It will have goblet cells in it, so it'll help you determine that this is the pseudostratified columnar epithelium. If it was ciliated simple columnar epithelium, what will happen is that the nuclei will all be in the same row, but you'll notice that the nuclei are all they look like they're stacked upon each other. Okay. If it was if it was simple columnar epithelium, all the nuclei will be kind of along the same row kind of thing. Okay, like this. Okay, but it's not like that, right? You can see that there are stacking here. Lateral surface means on the sides. What does the lateral surface of the epithelial tissue look like?
Hello?
I'm not quite sure what you mean by that. So can you?
Hello?
Yeah, so the basal surface would be the bottom, the apical the top. There's a question on the study guide saying the lateral.
Oh yeah. Okay. So here. Okay. So if we're talking about, so if this is a cell, right? If this is the cell, this is the apical surface, right? The one that's closer to the lumen here. The lateral surface are these on the sides of it. Let me reorientate. Let's go back to this one, okay? This one's easier to, since it's upright. Okay. So apical surface will be up here. Lateral surface on either side. Basal surface on the bottom. Does that make sense?
Yes, thank you.
Okay. Where are all these lecture posted? You mean the lecture videos? Is that what you meant? They are posted in the first module of your Canvas. It says lecture videos there. Yeah, you only saw two. The other one is still being processed right now. So stratified nuclei would be more spread out on top of one another. Correct, Nicole. They will be kind of on top of each other. All right. Okay. Even this, we could see another thing. Okay, let me quiz you on this now. If I were to point to, say, this tissue here, let me use a different color, I guess. This is a review. Okay, I'm reviewing something right now. What is this tissue right over here? Can't tell if it's one or two layers. Okay, well, hopefully we can help you. What kind of tissue is it? I'm not asking you that. What structure is that? All right, so let's take a look at this one. Okay, you could use this as an example too. They're the same. Okay, so there's a lumen here, right? See a lumen. This is the lumen. Right? See a lumen over here as well. And then if you look at this cell here, and looking at the cells next to it, right? Can you tell how many layers this is here? Making it a little bit more obvious, but hopefully you can kind of see what I'm doing here. Same thing as this, right? How many layers of cells are we looking at here? Yeah, we're only looking at one. Okay, good. And so what's the cell shape like? I'm going to take off the pen so you can actually see it a little bit. Where's the cell shape like? Yeah, cuboidal. Very good. So this is simple cuboidal epithelium. Okay, this is like one of the ducts that leads to the surface. Okay, good. Oh, by the way, I forgot when we were at this talking about this tissue here, we forgot to ask what organ do we have? What organ will have this tissue here? I'll come back to the how is it simple in a second, and why it's cuboidal in a second. Okay, so the respiratory tract, very good. The respiratory tract, which contains the trachea and the bronchi. Okay. The alveolus, unfortunately, is not going to be a pseudostratified. If you remember, for alveoli, what kind of tissue, epithelial tissue, is in the alveoli? What kind of tissue? Yeah, squamous. Which kind of squamous is it? Simple squamous epithelium. Okay, so for pseudostratified columnar epithelium, we'll have it in the trachea and in the bronchi. Okay, let's take a look at this back here again. If you look at it, it's only one cell layer. Okay, I'm going to circle around because it circles around the lumen.
Oh, okay. So also the bottom layer doesn't count. It's like all one big cell.
Yeah, it's one big circle basically. Exactly.
Oh.
It's one layer formed in a circle.
So then below that, the areolar connective tissue is
This
is
part
that
here?
one? Yeah, all that's one big blob.
Yeah, it's one big blob. Exactly.
Okay.
And you're right, because areolar connective tissue, we don't care about layers anymore since it's connecting things from the pseudostratified columnar epithelium to the other ducts or whatever this cartilage that we'll be looking at next kind of thing.
Okay, thank you. That helps.
Okay, yeah. This is how this is basically trying to train you to try to see these things. And it's cuboidal because if you look at the height and the width of these cells, they're more or less the same height and width of these cells. Okay. I didn't understand what the lumen did. What do you mean by you don't understand what the lumen did? This space here is the lumen. Um,
Oh wait, does she mean the space above it maybe? If you go to the right.
like here?
Yeah,
This
maybe
is one
that's
of those things that I was talking about with the tearing of the tissue. This space is an artificial space. There shouldn't be a space in here.
Oh.
Yeah. So it happens every so often. So this space does not mean that this here is epithelial tissue. Okay?
So
This is not epithelial tissue. And that's why I want you to start recognizing it, not only going by whether or not there's a space. That's like kind of the beginning part to help you recognize it. But eventually, I want you to actually be able to see what the tissue is and be able to recognize it just by the tissue itself. But the cell is still attached to the same base membrane. It's just in the circle. Exactly. The basement membrane is just in a circle. Oops, let me erase this. So the basement membrane is this right here. This is the basement membrane here. It's just in a circle.
I think what he's trying to say, I don't know if it'll make sense, but so the lumen is the hollow space of the cell. What's
not
happening
of the
is
cell, not of the cell. The cells are surrounding this space.
like it's surrounded, yeah. So then basically, it's still inside of it. It's just that the entire cell is surrounding it, but it's all the same base membrane. It's all the same cell. It's just around the lumen.
It's the same tissue.
Right?
I'm trying to the same tissue. Different. These are all different cells, right? This is one cell. This is one
Yeah,
cell.
there you go. It's the same tissue,
Yeah,
basically,
same tissue
just
that
around the whole.
surrounds the lumen. Exactly. All
Yeah.
this is the same tissue, correct?
So that all makes sense. I'm just confused about. Okay, so where's the basal membrane in that cell, and then where's the apical
Okay,
surface?
so the
then?
basement membrane of this is just the outer part here, the outline that you see here.
So there's no apical surface
The
for that
apical
one?
surface in this one would be over here. So the base is over here and lateral will be here.
Shit. Okay.
So the apical is where the lumen's located at.
Always? Okay.
Yeah,
Okay.
always where the lumen's at. It will be the apical surface.
And is it possible to not have a lumen?
Yes, there is a possibility where there doesn't have a lumen. And so that's why I said these are generalizing things. I'll show you the possibility that the one that doesn't have a lumen one, okay?
Jeez. Okie dokie.
It is confusing, but like I said, the more exposure you get to this, the better you'll be becoming. Okay, so
Fun.
and that's good that you're here. You know, and this is giving you more exposure of it so that you become more familiar with these things. All right, so with this one here, from here to here, what is this tissue here? Yeah, so many layers. Agreed. This is a lot of layers. Good. Okay. So you want to be thinking, well, first of all, is there a lumen here or is there a space? Yeah, we do see some space up here, right? Good. Now we see that there is a lot of layers, like someone said, a lot of layers. And then the next question is, what kind of shapes are these, right? Kind of thing. Well, what kind of shapes? Remember, where are we looking at when we're looking at shapes? We're going to be looking at the top, right? We're going to be looking at the top. The reason why we're looking at the top, because the bottom ones here, I would agree. This is kind of cuboidal, maybe columnar kind of cells. So you don't want to go by the bottom. You want to go from the top. From the top is very clear. The apical surface, what kind of cells are these when you look at the top here? Yeah, it's rather flat, right? These cells here, they're pretty flat. So this is stratified, like you said, multiple layers. Squamous, because the top layers, the apical layers are flat. And this is, since there's a lumen, it's an epithelium. Now we want to determine, Are these cells on top living or dead? Do you see the nuclei? If you see nuclei, they're living. If you don't see nuclei, they're dead. Do you see nuclei on here on the top layers? If you take a look, here's a nucleus, here's a nucleus, 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 nucleus. Okay? So these are living here. Okay? All those are living cells. And since they're living, this is going to be the non-keratinized stratified squamous epithelium.
Sorry, where's that word on the study guide?
Oh, it might have been grayed out, maybe.
I don't see it, but
Yeah.
I heard.
Don't worry about it. I'm not going to count it against you. Okay. It'll be like an extra credit kind of thing as well if I give extra credit out. Okay.
Okay.
But on lecture, you definitely need to know the difference between the two. Not by recognizing it, but by knowing by definition.
So pretty much if you can see like the dark spots, which are the nuclei, then it's always going to be, it's going to be living.
Yes, correct. Good. Now,
Okay, so if there was none of those like dark spots, then it would just be a blur kind of, and then they would be keratinized.
yes, yeah, very good.
Okay.
And
Just checking.
No, you're good. I'm glad you're asking these questions because I'm sure a lot of others are having the same exact questions as you.
Definitely.
Okay, so the top layers here, if you see the nuclei, that means it's living. If there is no nuclei, then they're dead. Okay. If it's living, it's non-keratinized.
That's the same thing I was going to ask. Do you have a characterized one?
Yes.
If it's not,
It'll be the next slide.
Okay. Thank you.
You'll see. I should have said that because now you know what the next slide is about. Anyways, talking about this tissue here, what organ do we see on this tissue? What kind of organ will be this this tissue? What tissue? I can't say. Where would you find this tissue? What organ will have this type of tissue? Yeah, the oral cavity, the esophagus, vagina. Not everything. Definitely not everything. The anus. Good. Okay. All right. So this next one here, if I say identify this tissue here, what would be your answer?
Would that one have multiple layers?
Yeah, it's multiple layers, right? You can see clearly there's a lot, just by even just this top part, right? You can see that there's multiple layers, but it also extends down to here as well, kind of thing. So we know it's stratified, very good. And there is a space here, right? So we know that there's a space, so we know it's an epithelium. Okay. Now let's kind of fill out, okay, what is the shape of these cells? Remember, where are we looking at when we're looking at these shapes? Yeah, we'll be looking at the top layers, right? And looking at the top layers, what are these shapes of these top layers here? Yeah, they're flat, so they'll be squamous. So my next question is, are these living or dead cells looking at the top layers here? Yeah, they're dead, right? You don't see any living cells whatsoever. There's no nuclei up here. So this is going to be the keratinized stratified squamous epithelium. Good. Okay, what organ do we see this at? If there are two of the same tissues, is it okay if we put the same example or organ or both? You're saying if they're two different tissues, but they're in the same organ, I'm guessing is what you're trying to say, Sadie? Is there this? An organ can have many different types of tissue. I don't know if that answered your question. Yeah, Ali, for now it's going to be a little bit tough, but the more that you do this, the better you become. Um, does that mean that they will be shed off? Yes, the dead tissue will shed off, correct. What about the bottom? We'll take a look at the bottom in just a second. Is the nucleus always found on the top, or it depends. If you look at this one, you do see some nuclei over here. You see a nucleus here, right? All these circling are the nuclei. Okay? All these darker staining structures are nuclei. When in doubt, put kidney. It's not in everything. It is in a lot, but not in everything. If we see two simple squamous, can we put kidney for both? Yeah. For the most part, you won't probably have a repeat of the same type of tissue, for the most part. Is it always skin? For the keratinized stratified squamous epithelium, yes, it is always skin. So that would be my next question. What organ will you find this in? So you're right, it would be skin. This is thin skin because there's not that many layers of the stratum corneum over here. Is that not for keratinized? I don't quite understand your question there, Noe.
I meant the nucleus, like like the nucleus is on the bottom, so like the keratinized stratified squamous will never ever have a nucleus.
Oh no. This entire thing, including this part here, is the keratinized stratified squamous epithelium. So they will have the living cells and also dead cells, both of them.
Oh,
And
thank you.
if you want to review what we talked about in the integumentary system, we could actually divide up all the different strata. Remember the come, let's get sunburn?
Yeah.
You could see all that right here. Well, sort of. Since this is thin skin, it doesn't have this one here, okay? So we have the stratum corneum, and that's this part here. This is the stratum corneum.
With the nucleus or no?
With the dead skin.
With the dead skin. Okay.
This darker part over here, where you have the darker staining parts over here, this is the stratum granulosum. And then all these living cells here with multiple layers, that's a stratum spinosum. And this very bottom layer here, anybody remember what the B stands for? Stratum basale. Very good. No dumb questions, correct. No dumb questions. Palms of the hands will have thick skin. Palms of the hand is thick skin. This is thin skin like the scalp. Very good. Like the scalp. Most of our body would be thin skin. Good. Okay, so some people were asking, what kind of tissue was this down here then? Yeah, it's connective tissue. Good. Okay, let's be a little bit more precise. What kind of connective tissue is it down here? How do we know it's connective tissue? Well, first off, remember, here was the space here, right? The space here, since there's a space here, what is the first tissue that encounters that space will be epithelial tissue. Notice how if we're going all the way down here, now there's a tissue in between the space and this tissue here. So this can't be epithelial tissue. So it has to be either connective tissue, muscle tissue, or nervous tissue here. Okay. So you won't have a lumen here with the connective tissue. Yeah, so for those of you who answered areolar connective tissue, that is correct. Remember, a lot of the times when we have epithelial tissue, we will have areolar connective tissue right underneath of it. Okay, most of the time we'll have that occurring. So whether it's pseudostratified columnar epithelium, like we saw earlier on over here, all this tissue here is areolar connective tissue. It's right next to the epithelial tissue here. Let's go back a little bit more. All this tissue over here, is right underneath the simple columnar epithelium, is areolar connective tissue. When we look at this, all this tissue over here between the renal tubules here are areolar connective tissue, kind of like over here as well, areolar connective tissue. Okay, so areolar connective tissue is usually associated with the epithelial tissue. Okay, all this down here, areolar connective tissue. So pretty much most of the times, areolar connective tissue is right underneath the epithelial tissue. Okay. Okay. Most of the time, not always, correct? Not always. So here, okay, let's see if you can figure this out. Okay, if I say what is this tissue here, I guess I will use a different pen color. That one doesn't really do the good job of it. From here to here, what is this tissue called? I guess I overextended a little bit, but kind of get an idea. Here to here. What type of tissue is this here? Okay, I see simple cuboidal epithelium for a couple people saying that. Yeah, is there more than one layer? Good question. Is there more than one layer? Do you see that? Same thing over here. I'm going to just repeat it. Okay. Is there more than one layer? Yeah, it looks like, yeah, I agree. It looks like there's more than one layer. So it can't be simple anymore. Okay. If it can't be simple, then we know it's stratified. Good. Stratify what though? Okay, if you take a look at these cells here, okay, what's the shape of these cells? Yeah, cuboidal, very good. Remember, I'm not going to test you on stratify columnar. Okay, that's not going to be one of the exam questions. Okay, remember how I've been saying, oh, is there a space? Is there a space? Is there a space? In this case scenario, it doesn't apply for this epithelium. This is not a space. This part is not a space. This is actually an oocyte, okay, which is a cell. So this is one of those exception that the epithelia doesn't have a space. But where you wrote is a space. Yes, you're right. Where I wrote is a space. But this is not, this will be a different type of tissue over here. Okay, this is a different type of tissue right here. Okay, this is a different type of tissue. But that's not where I labeled, right? I didn't label that, right? I label this right here. Right? If you follow this follows the circle, right? So you could even say right here. If it's right here, right, it's not facing this space anymore. If it doesn't face that space, then you can't say that's this is the lumen. If anything, you could say this is the quote-unquote lumen. It's not a lumen though because there's actually a cell in there. Okay. This is probably the only time that I'm going to have an epithelial tissue that doesn't have a space. Let's put it that way. Okay. This is probably the only time that I'm going to have a non-space epithelial tissue. Okay. Is the top part areolar connective tissue, all this over here? I think so. I'm not 100% sure, but I think so. It looks like it. We will not be looking at exact same photos on the exam. Hence why I want you to be familiar, keep looking at different slides online and whatnot. Use your textbook, use the Weebly site, and things like that. Yes, this is an ovary. This is an ovary. This is where we're going to find this stratify cuboidal epithelium. Okay, we also find it in sweat glands, and also in the testes, but this is the ovary. The virtual anatomy website, that's in your first module of your Canvas site. All right, I'm getting hungry, but let's finish up the epithelial tissue. Let's look at this one more slide, okay? So if I say identify this tissue here, what would be your answer? Okay, you want to think of those questions, right? Stratified cuboidal, that is a possibility. Okay, stratified cuboidal epithelium. Good. Okay. Stratified cuboidal. Everyone's saying stratified. Oh, here we get transitional epithelium. That kind of changes things around. All right. Well, let's first stop. There is a lumen, right? We see the lumen over here. How many layers is this? We do see that it is multiple layers, right? The nuclei are kind of stacked upon each other, so we know it's some sort of multi-layer stratified type of tissue. Good. Okay. What are the top cells look like? Okay. Notice how the top cells look like these. I'm going to try to outline them, right? This is what it kind of looks like, yeah? So what kind of shapes are these when we look at it? Okay, a little bit cuboidal. I would kind of agree with that, okay? But there was another one, right, that we kind of talked about. One that changes shape depending on whether or not it's filled with something, right? It is transitional, but remember how, yes, exactly. These are the clues that you want to be looking for, right? Transitional epithelial tissue. Not all the time, but every so often, there are cells here that are binucleated. And when you see this binucleated, and not only the binucleated, you can see that these cells are rounder in shape. And when it's rounder in shape, it's relaxed. How do we know if it's filled or not? I'm not going to give you one that's filled, okay? If it's filled, it will be flat. I'm not going to have you confuse that with the stratified squamous epithelium. Will the nucleus always be circular or oval? Yeah, circular, ovalish. Usually they will be. Not always, but usually. Will they take the shape of the cell? Not all the time. Remember how like the columnar and stuff like that, the columnar is a lot more elongated. Although the nucleus is elongated, it doesn't take the shape of it doesn't look like a rectangle. Yeah, this is transitional. This is transitional because of the binucleated cells, because of the rounder shape. Okay. So what organ is transitional epithelium in? Where would you find transitional epithelium in? What organ? Yeah, make sure you put urinary bladder. If you put bladder, I don't know if you're talking about the gallbladder or urinary bladder. Yeah, just make sure you say urinary bladder. Ureters, that brings the urine from the urinary bladder, from the kidneys to the urinary bladder. Does this have cilia? No, not in the urinary bladder. No cilia. All right. I think we're good, right? For now, at least. Let's get started on looking at these things. Make sure you review it. Start quizzing each other. If you have a study group, start quizzing each other. Start quizzing yourself. Like I said, going to that Weebly site, right? If you go to the Weebly site, you could kind of test yourself by going to tissues over here. Okay. And hopefully, you have it covered or whatnot, but there will be something pointing to something, and you'll be looking at this, and you'll be like, this kind of looks flat over here, so maybe a squamous of some sort. I do see a little lumen over here, not that much, but a little lumen over here. So, and there's only one layer. Okay, I think it's simple squamous epithelium, and plus it's asking for organs, so I would say this is from the kidneys, and then you double check. Oh yeah, I got it. Simple squamous epithelium, and kidneys is one of them. Okay, so you want to use this to kind of give you clues. The more you look at these things, the better you become, basically. Okay, so
Sir?
quiz yourselves. Yeah, go ahead.
So are we gonna have this exam on the 16th part of our lecture exam?
Yes,
Like, is it all?
you'll have it all in one sitting. So after you're done doing the lecture part, you'll do this lab quiz part of it.
Oh, so they're separate, just one's after the other.
I think I will probably keep it in one, so you'll have it like a one continuous exam kind of thing.
And how long do we have for all this?
I don't remember how many points I gave you for this section. I want to say 20 points, so they're probably going to be 20 questions.
Okay, just do we have time? I just want to make sure that we have time to answer because I'm slow and stuff.
Oh yeah, so typically speaking, for these lab exams, I give about 1 minute per question. When we did it in person, I would give about 40 seconds per question, basically. But since it's online, I might give you a little bit of more time, but just slightly.
Okay.
All right, keep it up, everyone. Study this, quiz each other, look over this, look over the lecture notes and everything. Look over your textbook. Use whatever you have available. Okay. And starting next week, CAS is open. So starting on Tuesday next week, start going to the CAS. Ask the tutors, hey, how do you figure this out? I can't tell between this connective tissue with that connective tissue. Can you help me out? And they will be helping you as well. Okay. Come to my office hours too. Okay. Keep coming. Keep coming. All right. Take care, everyone. Have a good lunch for those of you who haven't had lunch. I'm about to eat my lunch myself. Okay. See you.